This video is going to take you through the Microevolution Lab, which really starts on page 236 in the lab manual. It starts out by giving some information that we actually talked about in the first half of this course by just defining evolution and telling you a little bit about Darwin, but just as a refresher, evolution is change in a population over time. The first person who's generally credited with coming up with this idea is Charles Darwin, although remember there were people before him who thought populations changed over time. He is just one of the people who got part of the mechanism correct for how that population could change over time. He wrote about his findings in his book, which the full name was On the Origin of Species by Means of Natural Selection, but the lab manual just gives you the partial name that I put on this slide for you right here. The idea of natural selection is nature or the environment selects for certain organisms to be able to survive better, reproduce more, and their traits will become more predominant in the population over time because they were a better fit for the environment that was around them. Microevolution and macroevolution are slightly different from each other, but really only in terms of time scale. This chapter is about microevolution, and that's change over a relatively short period of time. We can see evolution happening in our day-to-day lives. It's just not something that the average person looks at. Last semester, well, it depends on, I guess, where we were last semester, but I usually do an activity that relates to antibiotic resistance. That's change that we've seen over the advent of just our lifespans, and so that means that's microevolution, antibiotic resistance is. We can also watch organisms that have a really fast reproductive rate, like fruit flies. We can watch them change over a relatively short period of time. However, organisms that take a really long time to breed, like human beings, it's a lot harder for us to see evolution on that time scale, and so that would be macroevolution, which is going to be the next chapter that we talked about after this. Now, population genetics is often used to study microevolution, and that's because you can start to see changes in the genes of organisms build up over a relatively short period of time, and so we tend to study those things since it's easier for us to see in our lifetimes or lifespans. So let me explain the picture that I have going on here. This is one of the examples of microevolution that we have seen within my lifespan, which I know you guys think I'm ancient, but I'm not really all that old. Fruit flies are these organisms. If you've ever had fruit in your house, you've probably had fruit flies flitzing around in your house too. They're annoying little bugs. They really like to eat sugars. Well, this researcher decided to take a population of fruit flies and then split them up into a couple of different jars. And in one jar, she fed these guys maltose, which is a particular type of sugar. And in a different jar, she fed these guys starch. Starch is a polysaccharide, which is a carbohydrate, but it's not a simple sugar. She then raised these fruit flies over several generations, feeding these flies the maltose stuff and these flies the starch stuff. And what they found over time was that enzymatically, these flies became better capable of digesting maltose than any other carbohydrate source. So their genes for the enzyme maltase became upregulated. These organisms on this side, on the other hand, they started producing more enzymes like amylase to help them digest starch, so they upregulated those genes. Then, if they tried to breed some of the maltose-feeding flies to some of the starch-feeding flies, they didn't like each other. They wouldn't mate with each other, and so we had actually created some breeding isolation between them where they preferred to mate with organisms that ate the same types of foods that they ate. So not only did we show the genes changed over time in terms of how they were regulated in the flies, but they also preferred to mate with organisms that ate similar sources to them. So that's a type of reproductive isolation that occurred, and again, we saw that within the lifespan of one researcher, so that's microevolution. And the next thing the lab manual does is it takes you just back over some terms, and that's because one of the big kickbacks we get when we teach evolution is that students like to say it's just a theory, and so we just want to remind you, a theory is not just a theory is something that has a massive amount of evidence to support the idea behind it. We have fossil evidence, we have genomic evidence, we have biogeographical evidence, we've got molecular evidence, we've got a lot of different types of evidence that support the idea that populations change over time. And again, we have it in the short term from antibiotic resistance to long term billions of years worth of fossils showing us how organisms have changed over time. So it's not just a They've defined all of these terms for you, but honestly, it's something that we talk about a fair amount in the first half of the course, so I'm pretty much just going to blow past that stuff. Now, they want you to just make sure you understand how hypothesis testing works here. And so they wanted you to do this little thing with a die, which is one dice. Dice is the plural. And so it said you have a dice on the table. I'm showing you a picture because we're not in a room together. You guys don't have a dice that's on your table. But it wants you to develop a hypothesis. What number do you think is on the bottom of this dice? So if the 6 is on the top, and these are 2 of the sides, and these are 2 of the sides, what number do you think is on the bottom? And then what color do you think is on the bottom? You can write those answers down towards the top of page 238. Then turn the block over. This is me turning the block over for you. Were either of your hypotheses correct? So did you know the 1 was on the bottom? In theory, you should have based on the 2 previous pictures and assuming it was a regular die. And then what color was it? Well, the other pictures were both white, so probably this was white too, and it was. And so in theory, your hypotheses were supported once we turned the die over and you were able to actually see for yourself what things were under there. After that, they have you wanting to do something else with a second wooden block, and I don't know what that wooden block was supposed to look like, so I can't do that one with you, so we're just going to skip the rest of page 238. But essentially, rolling a die and guessing what the number is going to be, that's a type of hypothesis testing. Okay, so now going back to evolution. I mentioned the term population earlier, so we need to know what it is. A population is a group of organisms of one species in one place at one time. They are capable of interbreeding. So like if you drive into a town, it tells you the population of that town. It's telling you how many people are in that town. It's not telling you about like all the people, the trees, the bugs, the dogs. It's just telling you about one species, which in the case of a population sign, that would be for humans. There are populations of dogs in the town. They just don't usually put that on a sign. Whenever you start thinking about evolution, it's very important that you understand that one individual cannot evolve. Only a population can evolve over time because evolution is defined as change in a population over time. And so it's important to understand what that population actually means. These 2 terms are throwbacks to last semester as well. They related to genetics. So the genotype is the actual genetic makeup of an organism. To use terms from last semester, organisms can be like homozygous dominant or homozygous recessive or heterozygous. We looked at things like flower color and the height of a pea plant when we were talking about these things. Phenotype is the outward appearance of an organism. While evolution does change the actual genetic makeup of a population over time, it actually acts by putting selection pressure on the phenotype of an organism or the outward appearance of that organism. Phenotype is not always just dictated by the genotype. Sometimes the environment can come into play there, and the environment can be anything from what kind of food sources were available, was there plenty of water available, were there any illnesses that were going around at a time. All of those can end up impacting the phenotype of an organism. Now, the next term, I prefer to call it allele frequency and not gene frequency because all the members of a population should have the same genes, but they might not have the same alleles of those genes. Remember that an allele is a different type of a gene. Like when we talk about this in the first half of the course, I usually throw out the example of eye color because that's what they teach you in high school. And so if eye color is a gene, then blue eyes is an allele, brown eyes is an allele. The frequency of those alleles in a population is something that can be static if the population is not evolving, or that frequency can be changing towards one or the other over time as a result of evolution. So what this is attempting to show you in this picture, it goes back to an example that we did last semester, which is these are pea flowers, and they come in either purple or white. They are doing W's for purple, so a big W is a purple allele, a little w is a white allele. If you count up all the alleles, you got to remember that each organism gets 2 alleles, one from mom, one from dad. So this one has 2 big W's, 2 big W's, 2 big W's, and so on for a lot of the purple ones. This one's heterozygous, so just 1 big W. And then these guys have 2 little W's, and that one has 1 little W. So if you wanted to count or figure out the allele frequency, you would have to figure out how many alleles you have in total, which is 2 for each organism. There's 9 organisms, so that's 18. And then how many big W's do you have? And when you count up all the big W's in the picture, there ends up being 13 of those. So 13 out of 18 tells you that 72% of the alleles in the population are big W, versus only 28% end up being the little w. So it is more common to have the purple flower in a pea plant than it is the white flower, based on this particular population in this picture that you're seeing. Now, natural selection is the process that can change a population over time in the real world without human involvement in it. There's three basic ways that nature can change a population over time: directional, disruptive, and then stabilizing selection. And I've got little graphs up over here to kind of show you what each one of those means. So if a population, let's say we're just talking about people just for fun. There are some people who are really tall. There are some people who are really short. But the average person ends up being someplace in the 5 foot something or other range, 5'8"-ish. If there's a type of selection that starts to favor people who are really tall or really short, but the medium-heighted people are selected against, that is disruptive selection. And so what would happen is the graph would end up looking more like the blue one if we do disruptive selection. Now we have the same population here: really tall people, really short people, medium-heighted people. If stabilizing selection were in play, instead, the really tall and the really short people would be weeded out, and we would shift more towards the midline. We would stabilize the population towards that midline. Directional selection, the red line really should have been the same as it was before. So again, we've got really tall people, really short people, and then medium-heighted people. In directional selection, what happens is, in this particular case, tall people are making more babies than anybody else because tall people discriminate against short people. And so the population would just get taller over time. And so you start to look more like one side than you do the other. The short people are dying out because nobody's having sex with them. The middle of the road people, they're getting lucky on, ladies' night at the bar. But it's really the tall people who are having the field day here. And so that's how directional selection works. So here's some other examples of each one of those. The lab manual talks about the peppered moth when it comes to directional selection. This is the classic example that almost all textbooks use in reference to directional selection. So this moth species, there's basically two different phenotypes that can be present. You can be a light-colored peppered moth, or they can be what's known as melanistic, which means they're really dark in color. Well, before the Industrial Revolution, there were a bunch of lichens on trees, and lichens are light-colored, sort of leafy-looking things, and so these light-colored moths blended into the lichens. That made it hard for birds to find them. The moths survived better. They made lots of babies. Life was all nice and good. After the Industrial Revolution, the air quality got so bad that the lichens on the trees actually died off. The tree bark became covered in soot from all the industrial burning of things that was happening in the cities. And because the lichens died off and the trees now had black bark, these guys started to do better because the birds could see the light-colored ones and were eating them. And so the population shifted towards the dark moth over time because the pollution ended up allowing them to hide better in the soot that were on the tree. So this is an example of directional selection. Now there's several different experiments that you can do. Your lab manual gives you one. They want you playing with beads and stuff, but I don't want you to have to go out and buy stuff you're never going to use again. If you happen to have beads, have at it. You can also do this with any kind of candy that's colored, like M&M, Skittles, Reese's Pieces, whatever. You still want to have the sort of paper or cloth, but you can do it with paper plates or paper towels or whatever. Run through the experiment that they give you, only instead of using white and black, you can use whatever color your candies happen to be. If you don't want to do that one, you can also type this link in right here, and there's an experiment that gets done fairly routinely in middle schools with teddygrams, where you get to see directional selection of happy versus sad teddygrams. So if you prefer teddygram graham crackers over candy, you can do the experiment with this. But this isn't something that I can do with you over video, and so you're just going to have to do that on your own. All right, disruptive. This is the one where the two extremes are being selected for, and the middle of the road ones tend to die off for whatever reason. So this time we're going to be looking at mice. I don't know if you guys have traipsed around in Texas very much, but in our area of Texas, we tend to have a lot of soil that is very pale. It's caliche, it's limestone. And we tend to have a lot of places that are shaded in or flat out burned over. If that happens, what happens to the mouse population is the ones that are very pale do well on the caliche, and so those numbers go up. And then the ones that are really dark, they can blend into the shadows or the burned areas, and so they tend to do very well. But these guys that are in the middle of the road, well, they're too dark for the caliche, but they're too light for the burned areas, and so they get picked off by birds and snakes more often, and so they become less prevalent in the population. And so what would happen is the population would shift towards those two extremes. Once again, there's a little experiment they have you trying to do in the lab manual. You can use the same candy sort of stuff as before. They also give you the example with oysters, but it's the same basic thing as the mice that I just showed you. So go ahead and do that experiment for yourself and you should be good to go there. Your last example is stabilizing selection. And my favorite example of this has to do with babies. So the normal baby size for a human being is somewhere around like 7.5 pounds. A baby that is this size has a head that is small enough to fit through the birth canal, but the baby is big enough that their survivability is okay. This is a newborn from Indonesia, and this giant baby must have just about killed mom in the process of trying to come out of mom. This is selected against because our birth canals are literally too small for a baby this size to come out, but we do have C-sections, and C-sections are what had to happen in order for this mom to give birth to this baby. But if nature had to take its course and we couldn't do surgery, mom would have died, baby would have died. That's why we don't have big babies off over here. Little teeny tiny premature babies, they also have health complications. Low birth weight babies are more likely to have lung problems, neurological deficits, blindness, just a whole slew of things that can go wrong with premature babies. So even though this would be a super easy labor and delivery, the baby wouldn't be as survivable. And so that's why almost all of our babies end up being around 7.5 pounds. That's the ideal. They're not too big, they're not too small. Mom is more likely to be okay, the baby is more likely to be okay. And so that's a really good example of stabilizing selection. They've got you doing yet another experiment with some of the candies. So if you're not diabetic, keep playing with candies. If you are diabetic, try to find some beads, I suppose. All right, after that, we've got mutation that gets discussed next. Let's see how far ahead I am in the book. Where are you, mutation? All right, page 248 is where the mutation stuff happens. Now, mutation is something that we also talked about in the first half of this course. It's a change in the DNA sequence. This time we're going to even elaborate on this a little bit more. So, mutations can be harmful or what's known as deleterious, which means you get that mutation and you are more likely to die as a result of having that mutation, or at least not be able to have babies if we're talking about it from an evolutionary standpoint. Mutations can be beneficial, which means they give you some kind of an advantage. As a result of having that advantage, you are more likely to survive and make more babies. Mutations can also be neutral, which means they don't do anything to you. They don't help you, they don't hurt you. Now, mutation is something that just happens because the enzymes that carry out DNA replication during the cell cycle, they're not perfect. They make mistakes. While we do have DNA repair mechanisms in place, those mechanisms aren't perfect either. So mutations do just accumulate over time. Again, we talked about this when we were talking about cancer and why you're more likely to get cancer when you're older. It's because mutations were more likely to build up the longer you lived. The same sort of thing works here though too. If you end up changing something that affects your phenotype and the environment either likes or does not like that new phenotype, you might be able to survive better or not, depending on how that phenotype is going to match you up with the environment that's around you. So basically, nature selects for or against mutations, depending on whether they're harmful, beneficial. Neutral doesn't really have all that much of an effect, though. So the example that I'm trying to show you right here, these are alligators. The black alligator is what's known as melanistic. Melanistic gators are really, really dark. They are much less likely to develop sun damage, which yes, gators can get sun damage, although it's not as likely to happen in them as it is in other things. If water is crazy murky, they can blend into that water really well. They can also do better if they're hunting at night because they're a lot harder to see. Albino, or there's another type that's called leucistic gators. These guys, they don't blend into the environment very well. So in nature, you will very rarely see an albino gator. And that's because, one, they're more likely to get cancer and die. But two, something's going to see them and eat them before they make it to the process of reproduction for themselves. And so this tends to be a harmful mutation that doesn't really exist in nature because nature selects against it. Melanistic is rare. It's more survivable, though, than the leucistic or the albino state. All right. Once again, they've got you doing a little experiment with some candy. You'll need a couple of extra colors in there, so you can do that experiment just so you can see how mutations can change the population over time. All right. The third process. Mutation was the second process. Natural selection was the first process. Gene flow is the third process that can change a population over time. This is really all about movement of organisms. Just because there's a population doesn't mean there aren't other members of that species that come and go. Like if you think about a pride of lions, the males come and go and move to other prides as well and take their genes with them when they go. And so like if we had this population of beetles and this population of beetles and this one decided, screw it, Mom, I'm leaving, I'm going to wherever, and he leaves and comes to this one, well, he just brought all his genes with him, which means the gene frequencies on this side changed because he brought new genes, or really alleles, in here with him. So gene flow is all about immigration and emigration. All right. Fourth and final thing that can change a population over time is genetic drift. And this one is just a luck or chance sort of a thing. Genetic drift has a much bigger impact on populations that are very small, which is why biologists get really concerned when we start talking about endangered species, because genetic drift can act on those species more easily than if we had a population that had really high numbers. Genetic drift is essentially something happened that even though there was an organism that was perfectly fit for the environment, it died somehow. This could be like a tornado came through and wiped them out, a fire came through and burned them, or the example in your lab manual is you stepped on them. And as a result of you stepped on this beetle, even though it was perfectly adapted by random chance, you stepped on it and killed it. You've now changed the gene frequency in this population over time just because you happened to step on those guys right there. So chance changes things, especially if the population happens to be really small. All right. After that, you have a whole slew of questions. Just go ahead and pause the video to get your answers. There's the whole first ones. Number 10, things get a little bit more complicated because they wanted you to do a little bit of math, but it's to make sure you understood how you could calculate that allele frequency. I feel like they have a numbers mistake on here because technically this adds up to 101, so I just rounded these off, but whatever, it gets you to round about the same thing. So this is the first table that's on page 253. They tell you that then we start a selection event, and then they give you a table at the top of page 254. This is what the table should look like on the top of that page. So they then ask, what color do you think the fabric was? Well, based on the fact that our survivors tended to be blue or black, it was probably blue, and it was probably a dark blue, because those are the ones that survived and reproduced. After that, we have critical thinking questions. In terms of directional selection, why do doctors recommend that you take all of your antibiotics and not save some for later? Well, directional selection allows bacteria to become more resistant to antibiotics over time. If you took all of your antibiotics as prescribed, you would have killed off all of the bacteria. So there can be no selection when the entire population is dead. Number 2, Hardy-Weinberg equation is used to calculate allele frequencies and tell if a population is evolving. How does it work? So Hardy-Weinberg is something that majors have to go into in a lot more detail. There's a lot of assumptions that go along with Hardy-Weinberg, but essentially it's just looking at allele frequencies, and if the frequencies are changing over time, the population's changing over time, which means the population's evolving. If, on the other hand, the allele frequencies are staying the same, that means the population is not evolving over time. So Hardy-Weinberg is a way for us to test whether or not evolution is occurring in a population over time. Number 3. Global warming is predicted to increase temperatures. In terms of microevolution, what phenotypes might be selected for or against in various plants and animals. So anything that is drought or heat tolerant is going to tend to do really well. We're already seeing expansions of things like mosquitoes. They're extending their range. Armadillos are traveling farther north than they ever have. So those things that can expand into these new places, they're going to be able to do that. Things that were cold tolerant, though, like polar bears, they've got no place else to go. They were already in the coldest place. The coldest place is getting much warmer. They are not adapted for that warmer environment. They're going to die off unless something changes. Number 4, how is artificial selection different from natural selection? So natural selection was nature or the environment selects for or against you. Artificial is we, human beings, select for or against you. Artificial selection is how we got all the different breeds of dogs and all the different breeds of cows and cats and things like that. So we have changed things over time. Nature can too. If you were to have read Darwin's book, the whole first third of his book is all about artificial selection, and he's talking about pigeons because back in his day, people bred pigeons sort of like today we breed dogs. And he was using that as his argument for, you guys know we can change pigeons. You've seen us change pigeons. If we can change things, why can't nature change things? So I really liked his argument for that. He was really trying hard to provide a lot of evidence to support his idea, and that to me is one of the stronger ones. Number 5. Why doesn't natural selection produce a perfect species that is adapted to every environment? Well, there's a lot of reasons here, but I'm just going to say it's because the environment never stays the same. The environment is constantly changing, which means we're trying to hit a moving target. So we're never going to be perfect because the target never stays in the same place. There is also the fact, though, that you can't produce a perfect species because there's so much chance at play. You have to happen to have the correct mutation in the correct gene to make you more perfectly fit. And the chance that happens is so very slim. So we're just never going to be perfect for a bunch of reasons. Number 6, what conditions have to be met to pass a beneficial mutation on to the next generation? Well, 1, you got to make babies somehow. Usually that's going to involve reproduction. If it's asexual reproduction, then that trait's definitely getting passed on. If it's sexual reproduction, though, that trait might not get passed on to the next generation because meiosis maybe didn't put it into the gamete that happened to get fertilized. And so we have to be able to reproduce and pass the gene on through the germline in order for that to occur. So that concludes your microevolution. Make sure you take the post-lab quiz.